Hello everyone, welcome back to the class. In previous class we learnt about glycosylation of proteins in Golgi and difference between glycosylation of proteins in Golgi and ER. And also we had learnt about the end-linked glycosylation and O-linked glycosylation. We had also learnt about how the structure has formed in complex oligosaccharide and high mannose oligosaccharide, the difference between both of these, and we learnt about how the oligosaccharide processing happens in the Golgi compartment. The phosphorylation of oligosaccharide on lysosomal proteins, that you will be learning today. This aspect will be learning today. And removal of mannose, addition of galnac, addition of gal, addition of nana, sulfation of tyrosines and carbohydrates. We all learnt it yesterday itself. So this is the end-linked oligosaccharide, and the difference between complex oligosaccharide and high mannose oligosaccharide is evident in this image. So a complex oligosaccharide is having how many N-acetyl glucosamine? N2 plus 3 is 5 N-acetyl glucosamine, that is what? Naga 5 NAGA, and mannose 1 2 3 blue color 1 3 mannose, and how many galactose? 3 galactose, and how many nana? 3 nana, that is sialic acid, 3 NaNa. So this was the resultant oligosaccharide after processing in Golgi, right? And this was the steps involving the modification of oligosaccharide taking place in the Golgi lumen. So let us quickly summarize. Oligosaccharide processing in ER and Golgi. So first in ER lumen, there will be removal of 3 glucose and 1 mannose. Before going to this, how many N-acetyl glucosamine and how many mannose were there? Just remind me. Yes, how many N-acetyl glucosamine, how many mannose, how many glucose were there? So to remember, there were 2 N-acetyl glucosamine plus 9 mannose plus 3 glucose, right? So here in first step, how many glucose are removed? How many glucose are removed in ER lumen? That is 3 glucose are removed, minus 3 glucose are removed, and also minus 1 mannose has removed in the first step. So let us count now. 2 NaGA is same, plus 9 minus 1 is 8 mannose plus 3, so there is 0 glucose left. After this step, the further modification happens in the Golgi. What happens in Golgi lumen? Again 3 mannose is removed. 3 mannose is removed. 3 mannose is removed and 1 NaGa is added. 1 NaGa is added. How many? Let's count now. 3 NaGa plus 5 MAN. 5 MAN. So remember this, 3 NaGa plus 5 MAN. 3 NaGa and 5 MAN is present. To this, there is removal of 2 mannose again. Removal of 2 mannose. Now how many? 3 NaGa plus 3 mannose. Both are 3, right? After that, in the last step, there is addition of 2 NaGa plus addition of gal, that is galactose, plus 3 nana. Plus 3 nana. How many are there now? How many are there? 5 NaGa plus 3 mannose plus 3 galactose plus 3 NaNa. Are you getting my point? So how many are there? I'll write it here very cleanly. 5 NaGa is present, 3 galactose is present, 3 mannose is present, 3 NaNa is present. So this is what the precursor oligosaccharide after processing in Golgi network happens, right? So next, what let us see now once. See, processing begins, removes specific mannose, remaining steps occurs in the Golgi stack. Then Golgi mannosidase removes 3 more mannose. N-acetyl glucosamine transfers transferase 1, then adds N-acetyl glucosamine. Mannosidase then removes 2 additional mannose. Here this yields final core of 3 mannose that is present in the complex oligosaccharide. At this stage, the bond between the 2 N-acetyl glucosamine between 2 N-acetyl glucosamines in the core becomes resistant to attack by a highly specific endoglycosidase. Then finally, additional of 2 N-acetyl glucosamine, 3 galactose, 3 sialic acids are added. Now today we'll learn about this aspect, that is lipid and polysaccharide metabolism. It's nothing, it's easy. Golgi apparatus functions in lipid metabolism. What all lipids does the Golgi produces or synthesize? That is glycolipids and sphingomyelin. You already know glycolipids and sphingomyelin are very much essential for plasma membrane. The components of plasma membrane are made up of glycolipids, cholesterol, integral proteins and extrinsic proteins. So these both are very essential for formation of plasma membrane. Ceramide, which is synthesized in the ER, is converted either to sphingomyelin or to glycolipids in the Golgi apparatus. Golgi is also a major site of carbohydrate metabolism. In plant cells, it acts as a site where complex polysaccharides of the cell wall are synthesized. Which are the polysaccharides or the components present in the cell wall? That is hemicellulose, pectin. Okay, in animals, most of the glycosaminoglycans of the extracellular matrix are synthesized in the Golgi apparatus. Now let us learn how the transport of proteins occurs in the Golgi. So before getting into this concept, I want you guys to make sure that we learned different types of transport vesicles, right? That COP1, COP2, clathrine, everything. So I'll write up flowchart now. From ER to Golgi to lysosome. From ER to Golgi, which type of vesicle is mediating the transport? From ER to Golgi, that is COP2. That is anterograde. Okay. From Golgi to ER, from Golgi to ER back, that is COP1. COP1. From Golgi to lysosome, from Golgi to lysosome, that is clathrine mediated associated with AP1. Clathrine mediated associated with AP1. So now we learn how the transport of proteins happens through the cisternae of Golgi. There are two models which explains the ordered transport of proteins through the cisternae. One is cisternal maturation model and another one is vesicular transport model. First, let us learn about cisternal maturation model. According to cisternal maturation model, stacks of cisternae are dynamic. Since most cisternae is assembled by fusion of transport vesicles from the ER carrying newly synthesized cargo. At each stage, the Golgi resident proteins that are carried forward in the cisternae are moved backward to an earlier compartment. So this model emphasizes on the retrograde transport. Each cisternae progresses into the next one and COP1 coated vesicles function to transport resident Golgi components in the retrograde direction. So in retrograde direction, which type of vesicles are helping? COP1. Next model is vesicular transport model. It says that a stable stack of cisternae containing a fixed and ordered array of processing enzymes are that cargo is delivered to each cisternae in turn by these vesicles that bud from one cisternae and fuse with the next in the stack. In this case, COP1 coated vesicles carry cargo and move in an anterograde direction from one Golgi cisternae to the next. Okay. Next is transport of proteins from TGN to the lysosome. So, which type of transport vesicle is helping from Golgi to lysosome? That is clathrine mediated with the adapter protein 1. Okay. So first, what happens? There is mannose 8 and N-acetylglucosamine. There are 2. First, what happens? There is phosphorylation happening. Phosphorylation of what? Phosphorylation of mannose. Phosphorylation, insertion or addition of phosphate group to the mannose residue, one or more mannose residue. In the resulting mannose 8 galnate 2 oligosaccharide of lysosomal enzymes becomes phosphorylated in where cis Golgi. The enzyme phosphotransferase transfers the phosphorylated N-acetylglucosamine to carbon 6 of one or more mannose. Enzyme phosphodiesterase removes the N-acetylglucosamine, leaving a phosphorylated mannose residue on the lysosome. What happens here? phosphorylation of mannose, one or more mannose residues happens. Especially the enzyme phosphotransferase transfers the phosphorylated N-acetyl glucosamine to carbon 6 of one or more mannose. So that result in M6P, that is mannose is M6P, mannose is phosphorylated in the carbon 6. Okay, first step is what? Phosphorylation of mannose and removal of N-acetyl glucosamine. Removal of N-acetyl glucosamine. So first step is formation of or phosphorylation of mannose in this carbon 6 and removal of N-acetyl glucosamine happens. Next step is there are some receptors in trans-Golgi network. There are some receptors waiting like this. There are some receptors waiting. That receptor is known as M6P receptor. M6P receptor. The name is evidently giving you a hint. What it receives? It receives M6P. It receives M6P. Okay. Many mannose 6-phosphate bind to mannose 6-phosphate receptor, which are found primarily in the trans-Golgi. The luminal domain of this transmembrane protein contains a region that binds M6P very tightly and specifically. So this M6P, just not M6P. So there is a transport vesicle. I'll write like this. I'll write in a different color. The clathrine coated clathrine AP1 coated transport vesicles contains M6P receptor. M6P receptor. This is a transport vesicle. It contains this M6P receptor. Okay. That M6P receptor is bound to what? M6P receptor is bound to M6P oligosaccharide with the protein or enzyme, you can say. So this will bind to each other. So where was this M6P? M6P is a oligosaccharide, right? Oligosaccharide linked to the protein or enzymes which is destined to lysosome. So this M6P is of the protein binds to M6P receptor present in the trans-Golgi network. This is surrounded by the vesicle, transport vesicle. So this has formed an association, temporary association. This clathrine, this transport protein or transport vesicle is made up of clathrine AP1 coated. Okay, clathrine AP1 coated vesicle along with the M6P and which is attached to the M6P of oligosaccharide of enzyme fuses with the sorting vesicle in the late endosome, which has an internal pH of approximately 5.5. The bound lysosome enzymes are released within the late endosomes. So this is our interest. This protein is or enzyme is our interest. Once it enters to the late endosome, okay, so it will fuse. So this transport vesicle first fuses with the sorting vesicle. After fusing with the sorting vesicle in the late endosome, this bound lysosome enzymes are released within the endosomes. Let us understand this process by the image. The addition and initial processing of the preformed end-linked oligosaccharide precursor in the rough ER is same for lysosomal enzymes as for membranes and secretory proteins. In cis Golgi, one or more mannose residues in the resulting mannose 8-galactate N-acetylglucosamine 2 oligosaccharide of lysosomal enzyme become phosphorylated. PGLCNAC you can see here. Enzyme phosphotransferase transfers a phosphorylated N-acetylglucosamine to carbon 6 of one or more mannose. Enzyme phosphodiesterase removes the N-acetylglucosamine GLCNAC after phosphorylating that mannose. It removes that N-acetylglucosamine, leaving a phosphorylated mannose residue on the lysosomal enzymes. Many mannose 6-phosphate binds to the mannose 6-phosphate receptor. See, binding to M6P receptor in the trans-Golgi network. The luminal domain of this transmembrane protein contains a region that binds M6P very tightly and specifically. Clathrine or AP1 coated transport vesicles containing the M6P receptor and bound enzymes bud from the trans-Golgi network and then fuse with the sorting vesicle of late endosome, which has an internal pH of approximately 5.5. So this transport to the endosome, these are known as transport vesicles. Clathrine coat, you can see here the green color one, it is a clathrine coat. The bound lysosome enzymes are released within the late endosomes. Phosphatase within late endosomes generally removes what? The phosphate. So first process after entering into the endosome is removal of phosphate, preventing their rebinding to the M6P receptor. So here when it enters, it dephosphorylates, removes the phosphate. After removing, the M6P and M6P receptor becomes separated. This forms two vesicles bud from late endosome. One type contains lysosomal enzymes but not M6P receptor. You can't see here the green color part, M6P receptor. Other type of vesicle is coated with retromer complex. It is coated with retromer complex. After these vesicles bud from late endosomes, they fuse with lysosome, delivers the lysosome enzymes to their final destination. Here you can see that enzyme has removed the phosphate, and this lysosomal hydrolase, which was the enzyme, is a precursor. It is delivered to the final destination. And here, what is the fate of M6P receptor? M6P receptor returns back to the trans-Golgi network to bring another set of enzymes into the endosome. So this is the transport of proteins which are destined to the lysosome. You can see the summary. First is addition of N-acetylglucosamine from ER. Mannose has come. A lysosomal hydrolase precursor which has labeled with the mannose is destined to the lysosome. It passes through the Golgi cisternae with many processing happening, and at last in trans-Golgi, till the trans-Golgi network, the protein or an enzyme which is destined for lysosome has M6P signal. With this M6P signal, it comes and binds with the M6P receptor. Binding to M6P receptor along with the clathrine or COP1 coated transport vesicles, it buds off and it is transported to the endosome. Right when they come inside the late endosome, there is removal of phosphate from the enzyme by the phosphatase enzyme, and there is dissociation of the M6P receptor from M6P because of the acidic pH inside the endosome. And the fate of M6P receptor, it comes and binds to the retromer coat. The M6P receptor in budding vesicle, the M6P receptor gets back to the trans-Golgi network to bring another set of enzymes, and the enzymes gets into the final destination. So this is the overall detailed image where there is what mentioned there has mentioned end-linked oligosaccharide with terminal mannose, UDP GL, that is N-acetyl glucosamine, which is responsible to phosphorylate the mannose. After that, the enzyme is mediating the transport and release of the lysosomal hydrolase from the oligosaccharide inside the endosome. So this is the summary signal sequence, Cadell sequence. Where did you learn? That Cadell sequence is responsible for protein targeting to the ER from Golgi. The vesicle type was COP1 coated. KKXX, where this protein is a membrane protein. The transport step is from Golgi to ER. The vesicle type is COP1. Diacid, which is having asparaginase glue, is a type of protein, membrane protein, which transports from ER to Golgi with the help of COP2 coated proteins, COP2 coated transport vesicles. Mannose 6-phosphate M6P is a soluble protein which is destined to lysosome from trans-Golgi to late endosome. The vesicle type is clathrine coated. In next class, we will learn in detail about the process of vesicle fusion, its type, and hypothesis which is related to the vesicle fusion. Thank you.